Good afternoon, Doc and classmates. I'm Elsa, aka Trixie Cruz, and we're going to present male reproductive system.
So first is testis. It is a compound tubular gland. It is a double gland because testis is both a cytogenetic and endocrine gland. It's surrounded by a thick connective tissue capsule, the tunica albuginea, that surrounds each testis. Lining epithelium of testicular ducts. Seminiferous tubules are lined by germinal or seminiferous epithelium. Tubuli recti is lined by cuboidal or columnar epithelium. Rete testis is lined by cuboidal epithelium. Ductuli efferentes is lined by an alternating columnar epithelium with cilia and cuboidal epithelium with microvilli. Ductus epididymis is lined by pseudostratified ciliated epithelium with principal and basal cell. Principal cells consist of stereocilia, which is a non-motile structure that absorbs fluid that helps the sperm cell to move. Basal cells, on the other hand, regenerates the epithelium. Ductus deferens is commonly known as vas deferens, is also lined by pseudostratified columnar ciliated epithelium. So next is spermatogenic cells. The 50 seminiferous tubule is lined by a complex stratified epithelium, lined by a basal membrane and covered by a sheet of connective tissue. The connective tissue contains fibroblasts and myoid cells. The myoid cells are contractile and produce gentle peristaltic waves in the tubules. The spermatogenic cells divide by mitosis, then meiosis to form gametes, which mature into sperm by the process of spermiogenesis. Unusually, the developing spermatogenic cells remain connected by cytoplasmic bridges until they have formed a mature spermatozoa. Therefore, spermatogenic cells undergo three processes called as spermatogenesis, spermatogonia, the spermatocyte, meiosis, spermatocyte, the spermatid, and spermiogenesis, spermatid, the spermatozoa. There are three types of spermatogonia. Type A, which is dark, which are reserved stem cells. Type A pale, which are renewing stem cells. Type B, spermatogonia, which are differentiating progenitors and form spermatocytes. Next is Sertoli cells, are the supportive cells of the testes that are located among the spermatogenic cells in the seminiferous tubules. They perform numerous important functions in the testes, among which are the following: physical support, protection, and nutrition of the spermatids, phagocytosis of excess cytoplasm from the developing spermatids, release of spermiation into the lumen of the seminiferous tubules, secretion of fructose-rich testicular fluid for nourishment and transport of sperm to the excrement ducts, secretion of hormone inhibin which suppresses the release of follicle-stimulating hormone from the pituitary gland, production and release of androgen-binding protein or ABP that binds to and increase the concentration of testosterone in the lumen of the seminiferous tubules that is necessary for spermatogenesis. Production and release of anti-Mullerian hormone that suppresses the development of Mullerian ducts in the male and inhibits the development of female reproductive parts.
Good day, I am Alessandra May Peñaranda and I will further discuss the other information of the male reproductive system.
So the interstitial cells of the lady, which are located between the convoluted seminiferous tubules, it functions as the stroma or supporting structure of the seminiferous tubule. It consists of Leydig cells that are used for the secretion of testosterone. Layers of seminal vesicle. Primary mucosal folds branch into numerous secondary mucosal folds. Secondary mucosal folds, anastomose, and form irregular cavities, chambers, or mucosal crypts. Lamina propria projects into and forms the core of the larger primary folds and the smaller secondary folds. Muscularis layer consists of an inner circular muscle layer and an outer longitudinal muscle layer. Adventia surrounds the muscularis and blends with the connective secretions of seminal vesicle. The secretions of the seminal gland have a key role in the functioning of the seminal fluid and make up 70% of its total volume. The various fractions contain mainly spermatozoa and prostatic secretion, while the fluids from seminal vesicles are included in the late ejaculate fractions, which contain alkaline fluid that neutralizes the acidity of male urethra and vagina for the survival of spermatozoa. Prostaglandins suppresses the female immune response to foreign semen. Clotting factors keep semen in the female reproductive tract post-ejaculation. Parenchyma and stroma of the prostate gland. The prostate is a collection of 30 to 50 tubulo-acinar glands embedded in a dense fibromuscular stroma in which smooth muscle contracts at ejaculation. The urethra that leaves the bladder and passes through the prostate gland is called the prostatic urethra. A transitional epithelium lines the lumen of the crescent-shaped prostatic urethra. Some of the prostatic glands contain solid secretory aggregations called prostatic concretions in their acini. The prostatic concretions appear as small red dots in this illustration. A characteristic fibromuscular stroma with smooth basal bundles mixed with collagen and elastic fibers surrounds the prostatic glands and the prostatic urethra. The prostatic gland produces a thin, watery, slightly acidic fluid rich in citric acid, prostatic acid phosphatase, amylase, and prostate-specific antigen or PSA. The enzyme fibrinolysin in the fluid liquefies the congealed semen after ejaculation. PSA is very useful for diagnosis of prostatic cancer because its concentration often includes in the blood during malignancy.
Good day everyone, I'm Snow White, aka Princess Brittany Abrigo, and I'm going to discuss the secretions of the prostate gland.
The tubulosinnar glands of the prostate are all lined by simple or pseudostratified columnar epithelium and produce fluid that contains various glycoproteins and enzymes and small molecules such as prostaglandins and is stored until ejaculation. A clinically important product of the prostate is prostate-specific antigen, a 34-kDa serine protease that helps liquefy coagulated semen for the slow release of sperm after ejaculation. Next is the prostatic concretions, also called corpora amylaceae, are small spherical concretions 0.2 to 2 millimeters in diameter and often partially calcified, and they are rounded eosinophilic bodies. Prostatic concentration are normally present in the lumens of many prostatic tubuloacinar glands and contain primarily deposited glycoproteins and keratin sulfate. Prostatic concentration may become more numerous with age but seem to have no physiologic or clinical significance. Next is the parenchyma and lining epithelium of Cowper's gland and its secretions. Cowper's glands, also known as the bulbourethral glands, are compound tubuloacinar glands. Each gland has several lobules with tubuloacinar secretory units surrounded by smooth muscle cells and lined by a mucus-secreting simple columnar epithelium that is also testosterone-dependent. These glands produce a clear, viscid, mucus-like secretion that during erotic stimulation is released and serves as a lubricant for the penile urethra. During ejaculation, secretions from the bulbourethral glands precede other components of the semen.
Thank you. That's all for today. Thank you for listening, and I hope you learned something from us.